And our next speaker today is Dr. Rachel Whitaker of the University of Illinois. She'll be talking about real-time evolution and gene flows. Rachel.
Okay, thanks. Good morning. It's been a great morning. I'm really glad to be here. I'm especially glad because of our morning speakers who really introduced some of the topics that I'm going to talk about. The first one was about evolution, which is really nice, as we all heard. But not only was it about evolution, but it was about how you can't no longer think of evolution as this kind of stepwise process going through time towards the ultimate pinnacle of the today's human, but also the idea that even the branching tree doesn't make sense, and there's kind of this reticulate evolution that you need to incorporate to understand how even the human species is evolving. So that's great because I'm going to be talking about that, but from a very different perspective. And then we had the talk from Shane this morning saying that talking about infection biology and individual heterogeneity, and I think that is going to be very clearly relevant to what I'm talking about today. So I am a microbiologist, and I think you guys are all catching on to, or you guys in general, the whole world is catching on to the microbiome revolution. That tells us that microbes are everywhere, and as microbiologists, we always have believed that they they rule the world. They are really important to health and to disease, and that the microbiome revolution has made us realize that they really are in us and among us all the time, and changing phenotypes of ourselves and the other animals and species that we care about. And that's going to be really important to think about and keep track of, especially in the context of what's being called now One Health, which is that the microbes from one human can move to another human. They can be shared between other environments. And so we're all kind of connected by this sort of web of microorganisms, bacteria and archaea. So I think that microbiologists in general have kind of lost this connection that's going on because they try to bring microbes into the lab and study them at a single individual level. So there's many studies that have looked at one clone of E. coli, how every single gene in it, how they regulate each other. But in truth, even the microbes are not an island in their environment. They are all also connected by a web of interactions. And that web of interactions is something we can think of as the microbiome's microbiome. These are mobile elements, plasmids, and viruses that are moving around and connecting individual cells to each other, just like the One Health idea of our microbiome is connecting us to everything else. So in order to understand the evolution and impact of microorganisms, we really have to understand how they live and change in the environment. So viruses get a bad rap because they're always thought of as pathogens, especially in microorganisms. They're mostly thought of as lytic, virulent lytic killers that can infect and then lyse cells. They're even called predators, or thought of as predators in the food web analysis and ecology. But actually, viruses are really important for another role, and that in not an ecological role, but more of an evolutionary role, which is that they can live within a cell. Every single organism that's been sequenced has mobile elements or remnants of mobile elements integrated into its genome. And when those viruses are latent in a genome, they can give their hosts new traits. So they're the source of evolutionary innovation. There's also chronic viruses that can infect without killing, and these also can come in and out of the genome. So we need to expand our view of these mobile elements and think of them as able to cause both this pathogen side, but also a mutualistic side that can give new traits to their hosts. And many of the viruses of microbes can kind of have multiple modes. They can be lytic or they can be latent. And so they can evolve along this continuum of symbiosis from antagonism to mutualism. And that will, of course, impact how the traits evolve in their hosts. So we're calling this idea infection genomics, and it's a kind of evolution that is important for us to think about, but hasn't really been incorporated into evolutionary models. Infection genomics is this incorporation of new DNA that happens through infection. So it's no longer mutation, but infection. And we think that this is really going to change the way we think about evolution. If you remember back to your population genetics, you basically need to know about a few things to understand how genetic diversity emerges and evolves over time. Mutation, selection and genetic drift, and then recombination and gene flow. Well, I think it's time for us to think about infection genomics as kind of an additional mechanism of evolution. It introduces variation just like mutation, except it's introducing a new genome with a whole composite of traits all at once. The variation can be moved around through infection processes where the interaction between the host and the virus or the host and the element is controlling where that infection happens. And then selection happens both on the individuals and on the combined unit, as well as genetic drift, meaning that this is truly a symbiosis. It's not an ecological process as much as it is an evolutionary process and the linking of those ecological and evolutionary processes of symbiosis. So I don't think we really know these rules yet, and so one of our ideas was to try to come up with figuring out these the rules for infection genomics. And this is a big problem. It's a big problem because we have to look in natural populations. And to really understand it, we need to do this in many different natural populations. So we need to understand how different types of organisms are evolving in sympatry with their mobile elements. To do this, we need to sequence populations of genomes. Cannot no longer look at this one individual clone. We have to look at big data levels of individuals on the order of thousands per type of organism we're looking at. And within those genomes, we have to recognize variation in the form of mobile elements and viruses, which are called the dark matter of microbiology. We don't really know what they look like. We don't necessarily know how to notice them, and they're rare. Every one could be different. Every individual could be different. And so this is, on the scale of thousands of genomes, needs some new computational tools. We want to look at it through both directions of a symbiosis, not just how the host is impacted, but how the virus is impacted and how those two, the fitness consequences of evolution on both sides of this interaction. And we also need to understand the traits that are encoded in viruses. Most of the time, if you find a virus in a genome and look it up, you don't recognize any of the genes that are on that virus, even if it's in a very well-characterized host. And so we need to understand how virus infection is impacting in a real molecular way the physiology of the microbes. So this is a really big, kind of broad question, and it also spans systems, and so it's hard to get funding to this kind of big question in evolutionary biology. And I'm very, very grateful to the Allen Foundation for taking a risk at trying to do this sort of an integrative look and giving us the ability to kind of go forward and address these problems in more of a holistic kind of a way. So I did mention that we need to look at it from both directions, and we actually got a really nice handle on that, a really nice tool for looking at interactions and virus-host histories with the discovery of CRISPR-Cas immunity. So CRISPR-Cas immunity is now really well known for its use in human, even genetic engineering, but it was originally evolved in bacteria and archaea, and it encodes a adaptive immune system. So just to remind you, if a virus infects a host cell, it at some frequency can add a new spacer onto this chromosomally encoded array of sequences in the genome of the host. So you can use this to look through time at the history of interactions between a virus and a host population by reading through that history and the memory that's handed down from one generation to the next. It also gives us a very obvious link between genotype and phenotype, because when there's a CRISPR spacer that matches a protospacer in the virus, the Cas machinery is guided to degrade that virus. And that means that we can tell, based on the CRISPRs that are in the genome, whether or not the given bacteria or archaea will be susceptible to infection, and what its history of infection has been. So being able to look at this in both directions has been a really useful tool to allow us to jumpstart this kind of answers to these big questions. So we were really excited about the fact that the CRISPR immunity gave us some of the tools and methods for looking at diversity and evolution that we have already been working on for a long time, with looking at human adaptive immunity. And many of the aspects of it are the same. There's diversity, memory, cross-reactivity, self-recognition. All of these pieces are built into this molecular system of CRISPR. So to tackle this problem, we took 3 different approaches. I'm just going to go through what we did in a model system. We took a theoretical approach in collaboration with Mercedes Pascal, who's looked a lot at the diversity of immunity and what it means about pathogen evolution in humans, especially in malaria and other organisms. And so we took a theoretical approach to try to understand what kind of diversity would be in the population. Then we looked at whether the diversity structure that we predicted from our model is in a very well-known model system. And then we are taking that into the lab now to see how the diversity of CRISPR immunity is impacting the evolution of a virus. So I'm just going to run through that really quickly right now. This is the model, the simulated data. At the top, you see the host genomes. We can track every individual genome, and the different colors are different genomes, meaning they have different CRISPR spacers in their CRISPR spacer array. And you can see that this simulated data, there's this really diverse set of hosts that evolve and persist over long periods of time. But then at some frequency, there's this plummet in the population size and a rise in the population size of the virus. So there's these epidemic outbreaks of virus that are kind of punctuating this stable diversity through time. And with Mercedes and others, we decided we wanted to look at what happened in the transition between these different regimes, between stability and this kind of epidemic outbreak. So to do that, we took a network-based analysis. We didn't just look at counting the number of alleles, but because different alleles can match multiple individuals, we needed to take a network-based approach. And you don't have to understand all of this, but basically what we did is linked hosts and viruses to each other through their spacers and protospacers. Remember, where they match is where immunity is conferred, and so we can put those together and see how hosts match viruses, and then we can construct matrices that are going to tell us, make us able to predict the infection network, where viruses can infect hosts, and the immunity network, where hosts are immune to viruses. And we thought that if we understand these interaction and immunity and infection networks, we might be able to understand what causes these shifts between regimes. So here is a movie that is going to show how the immunity network changes through time. So this is all of the host genomes and all the virus genomes, and where they match, you can see the colored box. And the colored box is telling you how many times they match that genome. And so you can see that, especially in this stable wall time, there's very few viruses, many different hosts, right? And the viruses are actually going extinct. They're kind of, they're matched so many times that they can't find a mutation that allows them to access a new host, and so they die off. And as they die off, the ones that are left have room, left over have room to grow on a few hosts, and those are the ones that are matched by very few spacers. So we see this really nested network that is basically taken apart. She calls it peeling the onion to make very few strains. And then as we come to this next outbreak, you'll see that the viruses expand and the hosts expand. And as they're doing that, they're building immunity, where these red boxes are increasing. So you have more immunity that builds, and then you launch into the next regime. So we've done some analysis trying to figure out what causes this kind of complex behavior, and we think that it's really important memory. The length of memory is really important, especially as it relates to the mutation rate of the virus, the viral persistence, the length of time that viruses can stay in a population and how diversified they are, and also their competition through cross-infection. Okay, so now moving to the data, we used a model system called Pseudomonas aeruginosa, and in this model system, it's an infectious pathogen, and it is well known for having a big genome that can accumulate all kinds of mobile elements. This is an epidemic strain, which is probably the best characterized natural clinical strain, and it is infected by multiple different viruses, and these multiple different viruses impact its fitness in the environment and make it able to be epidemic. So the viruses play a really important role in the trajectory of disease in this system. So we decided to take a look at how CRISPRs and viruses match each other. And to do this, we used a global data set of published strains. And we looked within a single human, chronically infected human, and between individuals to try to look at the diversity. And we saw that within a human, there's no CRISPR diversity, but between humans, there's lots of CRISPR diversity. So we took those CRISPR spacers and we compared them to published genomes of viruses or phage that infect that Pseudomonas strain, and there's many different published genomes. We put them into families based on how they interact with the host. Remember, you can be lytic, or you can be latent, or you can be both as in a temperate virus. And we found, surprisingly, that most of the CRISPR spacers that we found matched to a certain family of temperate viruses. So they did not match the lytic viruses as you would expect, but they did match the temperate viruses, the ones that can either be latent in the genome or lytic. And some of them were matched significantly highly in the population. In fact, when we looked at it, we saw that it is a nested network. It's modular in that there's certain spacers that match certain viruses, which is something we predicted in our model. And that modularity, we think, is leading to this nested network, which you would expect after an outbreak from our simulation. So that led us to wonder what's happening in this population. And we go back to thinking about, well, there's two different lifestyles. How would a virus respond? What's the virus view of this diversity? How would it respond? And the prediction is that if there's no new hosts because there's immunity that's stable in the population, and you have the option of staying with your host, that the symbiosis would evolve towards a mutualism between a latent virus and its host. And so we wanted to know whether that would happen in our system, and so we set up to do this in an experiment where we had an infected host and a CRISPR positive host, and we incubated them together Oh, sorry. We looked out in the, I forgot, we looked in the 1,000 genomes and we found that in fact there were many infections of this type of virus in the native population. So it is a strategy that this virus takes, is to integrate into the genome. In fact, 53% of the, or 56%, sorry, of the genomes, 1,000 genomes that we looked at were latently infected by a temperate virus. So here's our experimental data. We evolved it over time, and this actually took some really, it was difficult to do. We did this using deep sequencing approaches, and we found that CRISPR alleles did diversify over time, but really what happened is that the infection spread in this population. So they did take this latent approach, and these are all new infections. We can tell the difference between new and old infections, which is something that's really new and different. So in this context, in fact, they do take a latent strategy. So, as I just said, we have figured out that the environment that the viruses and hosts are interacting in is changing their interactions and selecting for a style of a life cycle, which is to integrate into the genome, and we're thinking about this in lots of other systems. So our group has, the funding from the Allen Foundation has allowed us to put together a group of diverse backgrounds to look at these questions in many different systems. And as you can see here, we have modelers and we have engineers and we have microbiologists and we have many different people that we can all put together thinking about these same questions. And we're together trying to put it into a bunch of different systems. Somehow my slide, okay, which are listed here: plant symbionts, antibiotic resistance. We're looking at swine populations. We're looking at the Pseudomonas populations I told you about, a diversity of systems in which viruses make a big difference to the evolution of the microbes that inhabit these systems. And this is my group at Illinois, and thank you so much for the funding that allowed us to do this.